can see. Okay, can you see my screen?
Yes.
Okay. Today we will talk about the network biology or biological networks. And as you know, there are many kind of networks in in the different part of the nature. For example, you can see different interaction between organisms, for example, in trophic chains, and when they are sharing nutrients or when they are sharing and creating energy and sharing energy, and many of these networks, particularly in biology, have direction. Sometimes they have magnitude, and sometimes they are able to follow auto-regulation or changes. So there are many examples. And at the end, why we want to do this kind of analysis and to see this kind of description of the interaction, because in terms of system biology, that is necessary because many of the information that we can see, for example, in different experiments or different analysis, is just one part of the big system. The enormous amount of information that we have, for example, in one organism like a human body, or in kinetic of growth of bacteria or something like that. There are many kind of data that, if we are focused particularly in one kind of data, we are just looking for we are just be able to see a super small amount of information that sometimes is not able to reflect the whole amount of information that we have. And sometimes it's difficult to understand the whole process that are happening, for example, in the cells. That is a very nice example that I am showing here, that I don't know that you know about this. It's an Indian history about the elephant, a big elephant in the room, where many people is trying to see they are trying to know without see the information, just touching a small part of the elephant, and some of them can say, okay, this is a snake, or this is a tree, or if somebody is touching the ears, they he can think, okay, this is a fan. So that means that we have good observation, because it's a good observation, even it's super directly observation, but not necessarily is the whole information, right? So in this class, we will start working and trying to understand biochemical reactions and biological networks related to signal production, metabolic biochemical reactions, and some interaction protein-protein, and another kind of network related to this kind of experiment. And in the last part, we will talk about gene expression and this kind of network that we can get from gene regulatory networks. So we will see some examples, and we will see a little bit some equation related to this kind of networks. So one question that we can see and we can ask us is, how is it possible that a cell with thousands of genes can make current decisions, like responding, for example, stress, dividing or differentiating? How can do that? Some comments from you? How a cell that is, for example, in bacteria, that is a single cell, can do everything? Is not necessary for one gene, right? Or two genes. It's many genes that are able to take a decision, like a thinking, right? How they can do that? How the cell is able to fix all of this stuff and resolve this big complex problem?
Well, I think it's because of evolution,
Okay.
but even taking that into account, I believe it's really impressive because we are talking about like compounds. things that are not alive per se, but they are doing like actions, very specific actions, uh, driven by interactions, chemical interactions.
Yeah,
But
okay. That is true. That is totally true. And these interactions, there are many genes, many molecules, many compartments, and all of them are interacting, right? All of them are working in different moments with timing, because timing also is very important. Where is happening? What kind of information they are processing? And they are able to do that because they have a real network happening at the same time. Not like a taking decision, not like a thinking, but just like a processing and changing some flux of information with very nice, uh, like a consortium, this is very clear that it's happening in the cell, and it's just not one gene or two genes. For this reason, during a long time in classical biology, we were working a lot with, for example, a function of one gene that I can mutate, or the protein that I can do purification of the protein and to see some characteristics. But maybe the real phenotype that I want to look for, or the real, for example, in case of medicine, when we are looking for some characteristics in illness or something that we want to create a new treatment, maybe just looking the information of one gene is not enough. So during a long time, we accumulate this amount of information about, for example, one gene that is able to produce some compounds, and this compounds is very important for another stuff. But in this moment, we didn't have enough information about that multiple layers that is able to happen at the same time in the cells. For a reason, now in the current biology, the real way that we are follow this information is as a system in system biology, right? And one of the tools that we are using in system biology is to understand this interaction using networks, and particularly graph networks that we can put in one single graph and to show super complex interaction. This kind of graph, for example, is like this one, like you can see here. Let me put my pointer. Okay, let me see if I can put. Okay, you can see here like these kind of figures. What is this stuff? What is suggesting this graph? You know,
Are they genes?
could be genes, could be proteins, could be many stuff, right? But in general, do you know how we describe these kind of points in this kind of plots? They are. This is our nodes. Our node, this is central point that have connection with another dots. The nodes are described like whatever kind of uh, element in the cell. Can be proteins, can be a transcript, can be a gene. And also we have this kind of connection, right? That are our age. And this is a general equation that describes one graph at network. That is, the graph is equal to the nodes and the edges. And all of the edges mean something, right? We will see what mean all of the edges. But these connections are important because we are describing how the different elements are interacting with another elements, and how they are forming different clusters, for example, and how they are forming different futures, done more than the future that we are observing just when we are watching one gene. So here, in general, we can say that a biological network represents a complex web of interaction among biological molecules like DNA, RNA, proteins, metabolites. And this is a different and primary language for understanding how the life process are orchestrated at the molecular scale. So nodes are biological entities, and age are interaction or reaction between them. Most of the networks are incomplete and difficult to interpret due to the relationship complexity and data peculiarities. So many networks can be super interesting for one kind of stuff that are happening in the cell, but we can complement with another networks. For example, we can complement transcription regulation and protein interaction, and are different kind of information that we can try to complement together to understand something. So we can finish when we are trying to understand something about complex process in the cell, we can have many kind of network working at the same time and trying to understand what is happening in general. So network visualization is a fundamental tool for uncovering important properties of biochemical process. This is a very nice tool because we can summarize all of the information in one single graph that we are able to understand. And this kind of network have properties, particularly biological network have very nice properties. So what kind of network we will see? There are at least 4 kinds of networks. Signal and gene regulatory networks that are the coordination between the response to stimuli and control of genes activation or repression. Protein-protein interaction, PPI, there is a physical interaction between the proteins. You know that many proteins are not working alone. Even some of them just are working when they are forming a complex. And these complex are super important to know if something is necessary in some context of the cell that is necessary to happen. For example, some transcription factors need to be with another protein together to be able to detect the region of the DNA that they need to regulate. So there are many interactions that are important for many functions, and we can understand this interaction creating this kind of plots, protein-protein interaction. We know that they are together, and we can put a graph with the different nodes and the edges, you know. So another network that is very common is metabolic networks. That is talking about enzymatic transformation of metabolites, energy production, and biosynthesis. And the last one is a phylogenetic trees that also is a kind of networks that is talking about how similar are genes or protein between them. And you know, this is a part of evolution topic, but it's very important that you need to know that this is another layer of we can represent in networks because they are connected by similitude between genes, sequence, and the different species that we can describe and how they are having connection between them. So this is the kind of network that we can have, at least in biological networks. So what is our molecular players? DNA, and just remembering this part is DNA is about the information, genetic information, store the whole information of all of the proteins, and this DNA is transcribed into RNA. The RNA is a intermediate messenger, you know, is producing in the nucleus, and they can go to, at least in eukaryotic cells, go to the ribosome to produce proteins. But there are another kind of RNAs, for example, Myrna and Sirnas. I don't know that you know about these kind of RNAs. Somebody has information about that?
For for silenciar genes. I just remember that.
Yeah. That is true, but you know what kind of RNA are? Because the RNA, for at least in eukaryotic cells, the RNA mRNA is the whole gene with the whole information of transcript starting with the codon that is ATG and the whole information about the introns and exons. But you know what happened with this kind of mirror RNA? What is the main characteristics? Just to remember.
No, I don't remember.
Somebody more? Any else?
Are they like short sequences of
Exactly,
RNA?
exactly. They are short sequence and they are not codifying, right? They are not codifying for DNA, for genes. They are not genes, they are not proteins, they are regulated elements of regulation in the cells. As Daniel said, they are silencing genes and they are super short, around 21, 22, 25 nucleotides. And they are crossing the different barriers and trying to regulate negatively the DNA expression, the RNA expression. You know, just is remember some stuff about that. And they are no usually they are can be in intergenic regions, but also they can be producing in introns or some uh, region of no heterochromatin, no codified. The another kind of elements that we can see are proteins. The proteins have different functions, for example, enzymatic functions, but also structural function, transport, signaling, regulatory, like transcription factors. Transcription factors are very important in many kind of networks, at least in regulatory transcriptional networks. We will see that, but also it's very important for metabolic networks, the enzymatic changes. And there are some classical description of enzymatic in the different reactions in the networks, and we will see a little bit about that and how we can represent that in networks. And the last one is the metabolites. For us, it's very important to know that we will have some products, some compounds that are processing by enzymes, and after that they will produce something. But also at the same time, there are small molecules that are participating in this interaction. For example, ATP, ADP, NADH, and amino acids, and they are able to take these small molecules and together with another molecules and create new products during the transformation, metabolic transformations. And that is very cool to follow that in some plots, in some graph. And we can represent all of the reactions in some of these kind of plots. This is just remembering a little bit about the transcription. You know that transcription is happening in the nucleus. This part is processing by RNA polymerase 2, and the RNA polymerase 2 is processing all of the information here, and they are producing RNA, a single chain, and this single chain is crossing the membrane of the nucleus, and after that they start to produce proteins in the translation process in ribosomes. So this is very simple example that how they are correlated information that is in the gene and information that is in proteins that are able to do something new. So we will see the first part that is very important for us, that is understand signal transduction and what networks we can create starting with this information, signal transduction in response to stimulus. This kind of interaction start with external signal, sometimes happen internal signal. We will see some examples about that. But usually there are some external signal like a hormone, growth factor, environmental molecules binds to receptors in the cell membrane, and these receptors are changing something like, for example, modifying phosphorylation reaction. And after that, there are receptor activations by ligands binding activates. And these receptors started signal cascades. Do you have some idea about one example of signal cascade? Typically, what kind of signal cascade we can find in the cell? Any idea?
I remember one that is related to inflammation processes, starting with TRPV1.
Okay, nice. Another example that you remember, for example, processing metabolic process, simple metabolic process. Insulin, for example, response to insulin, response, even the response to different hormones start with some receptor in the membrane. And after that, they start to a super similar process to kinase cascades. We will see a little bit the kinase cascades, but in general, these signal cascades are usually sharing phosphate during different steps of the reaction where they are losing some phosphate and they are transferring this information until some transcription factors. And transcription factors, when they are activated, they can start transcription or inhibit transcriptions with bind directly to the DNA in some regulatory regions. What's the name of these regulatory regions? Do you remember the name of these regulatory regions in the gene? Do you remember where the transcription factors are binding?
To a promoter?
Promoters, exactly. Exactly. This is the region that the transcription factor detect, and they start the transcription because they are activated. So just I want to show you something here that is transcription factors usually are activated by cascades, final cascades that are, for example, changing the phosphorylation state of the protein. And after that, they go to the, even many transcription factors, they are not able to cross the membrane of the nucleus because if they are not with phosphorylation state, they are not able to cross the membrane. So this signal is able to promote that they are crossing the barrier and after that starting the transcription process. Do you have idea how long time take this kind of process, like detect the signal and after that start the transcription of the genes? Do you have any idea about that time? Do you think that is 1 hour?
I don't know, but I would say that is seconds or even less than seconds.
Okay, you are telling me that the cell is able to detect one signal and after that phosphorylate and after that phosphorylate the transcription factor cross to the membrane of the nucleus. They look for the region, a specific region in the genes that we have ton of genes, for example, in human, there are around 30,000 genes, and they look that region and they detect this region and they start the transcription factor, the polymerase start to polymerization across the whole region of the gene, and after that they produce the mRNA, and they go to the ribosome and produce protein. Do you think that is seconds?
At this point, I wouldn't be surprised if it's less than 1 second.
Right, right. That is true. And also, it's true that it's in seconds. It's in seconds. It depends on the timing regulation because, as you'll say, the revolutions give some rules, right? And some stuff needs to be faster than another one. You know, for example, response to light is super fast. We know that some repair DNA proteins after to perceive the light, they start a transcription super fast because they need to repair, they need to protect the DNA. So these kind of reactions are around seconds, and another are a little bit more slow, but it depends about the regulation in general of the cell. But it's true, it's super fast. They are detecting and they are producing proteins super fast. That at least you can detect genes, mRNAs in seconds in some of the genetic activations. And it's real that is super, super impressive, right? So And at the end, we can see gene response. That is, some genes that are repressed or activate by the transcription factors. And after that, they are able to produce proteins, you know? So this is the phosphorylation chain, the phosphorylation of cascade of different MAP kinases that they are perceiving something in the membrane, and after that, using ATP, they phosphorylate, and after that, this ATP give a phosphate to the PK, and after that, they activate another PK with this phosphate and share this phosphate, and at the end, you can finish with a MAP kinase with many phosphates. And with this energy, with this molecule, we are able to activate transcription factors. So here I want to show you that here we have a network, right? And we have a connection here. And we have a connection with a transcription factor. And we have a connection that many transcription factors can activate another transcription factors. And these transcription factors also are activating another genes, you know? And we are starting to see that is starting a very nice connection between the genes and between the proteins that we can describe in some graph. So how we can describe that? We can say, okay, we have a signal, and this signal activates a transcription factor, and this transcription factor can go to the promoter of the genes and start the expression of many genes. Many of them also in another level of the complexity, they can activate many kind of transcription factors, and they can co-regulate many genes at the same time. And at the end, we can see the result of all of this information, because also you need to know and you need to remember that not many stimuli come from alone, right? If you are, for example, in nutritional starvation, but at the same time you are in pH, low pH, this reaction is different. The perception of the environment is different, and the reaction in genetic changes is completely different. So all of these information are happening at the same time and are processing at the same time for that cell. So it can be a little bit more complex than we can expect, right? For example, here, this is an example in the cell, like a signaling process, where we can see growth factors, chemokines, hormones. We can see, for example, some cytokinins and death factors. And all of them are processed by many kind of mechanisms. For example, you can see here genes like a CDC, that is control of the duplication or cell cycle. And we can see, for example, here how they are promoting, for example, cell proliferation. But this information here is super complex. And this is just one process that is about growth. And at the same time, is happening, for example, nutritional changes or changes about the response to light, for example. So that can be super complex, represent all of our interaction with this kind of graph, it's a big model, but it's super difficult to understand something here when you are watching all of the stuff at the same time. So for this reason, in system biology, we try to see this representation in simple graph. Like, for example, here, you can see the same stuff that are happening in the plot, but with external stimuli represented by some inputs. And after that, the direction of all of the stimuli. And after that, all of the region that where are happening, all of the reaction, and what kind of metabolism are starting. So we are reducing the complexity of this network that is happening in the cell, trying to understand more information at the same time. For this reason, it's necessary to know this kind of representation, because we have a lot of information and we need to see everything together, and with us summarizing all of the stuff, to understand something new, not just that one process, if not many processes that are happening at the same time, to understand what is the final reaction or the final stuff about that. For example, growth process is very important in cancer, and sometimes everything is happening good, but maybe when you are exposed to UV, something change completed, and you need to understand what is changing in these reactions. And for example, if something here in the metabolomic process, for example, respiration is something changing, and you need to detect these changes, that is not obvious, right? We are trying to see emergent information, know about one gene or about one pathway, if not emergent characteristics from the system. So we have some kind of network. We are talking about gene regulatory network that is about this relation between the DNA, RNA, transcription factors, that response to the stimuli. And we have this model that is graph equal to nodes and edges. And in this case, particularly in regulatory network, we are talking about genes and interaction of the genes. So there are another kind of stuff that we can consider. For example, cellular compartments, like if something is happening in nucleus, or something is happening in cytoplasms, or something is happening in another organelle, and we can put this information in our network. What kind of information we can find in for example, in different databases? For example, we can look for TAGG, that is a very nice database for pathways, or transpath, that is about also metabolic pathways. And the idea is to understand the regulation and to show the information in one simple graph. For example, here you can see the regulatory network of E. coli. This is a hierarchical layout. You can see, for example, this protein is affecting this set of protein, but also is affected directly to these kind of proteins. And here, for example, you can see that they are supercentral element. What characteristics you can see here? Super fast. What characteristic? What new information we can abstract from this information? For example, what is obvious here? What characteristic for you say, okay, this is something that is maybe in something unexpected, or something that is interesting to observe? To me, for example, look, this one are genes that are a little bit disconnected, but there are some of them that are super connected, right? This is a characteristic of biological networks, that some of elements are not super connected, but another one are super important. Another set of characteristics is that apparently there are like a different general genes. They are like a master genes, and after that, some of them that are not central but are super important to follow the information. So there are different hierarchical uh, stages of the genes, that some of them are super important, some of them are important to connect with another genes. For example, these genes is not able to connect with these genes because they need to be connected with these genes that are important for the flow of the information, the flux of the information. So there are many characteristics here. We will see all of these characteristics that we are observing has a name, and all of them are part of the math behind of these networks. So we will start also now with protein-protein interaction. Somebody has information about protein-protein interaction, how we can collect this information and how we can understand this information, and what kind of experiment we need to do to understand or to get this kind of information about protein-protein interaction. Karina, do you have any idea, any comment?
I don't know. I was thinking about molecular docking, but I'm not sure.
Molecular, let me,
Docking.
okay, between them.
Yeah, like in silico first to predict kind of possible interactions, but I'm not sure.
Yeah. But this is more directly is about in real interaction, physical interaction. How we can know this in physical interaction? Do you have any idea, Paula? How we know the
No,
physical interaction?
I don't have like an idea.
Yeah. Okay. Networks starting with physical interaction talk about real interaction in the cells, like forming complex, or even when they are working together in some process like transcription of genes. Polymerase sometimes is together with transcription factor, and they need to be together to be able to turn on a specific gene. Okay, there are many of this stuff. So the networks are similar to the regulatory gene networks, but they have another characteristics that is very interesting. That is, they are directed interaction. So we can know if they are interacting directly. We are not talking about if they turn on something. If not, they are talking about if they are touching, and after this moment they are or activating or inhibit or just giving phosphorylation. You know, this is kind of information that we can see here. And how we can know this information? There are mainly two techniques. The first one is GIS2 hybrid. You know about GIS2 hybrid? This is a super, it's old technique, but it's super nice technique. This is the way that we can know how they are in if the protein are or not interacting. And we can create networks starting with this information. Do you remember something about GIS2 hybrid? This is very cool experiment. You know, the problem is that for example, imagine that I have a protein that I don't know nothing about this protein. And I have another protein that I know something. But when I have the expression of this protein, or when I am starting expressing one protein, I am not able to get the phenotype. So maybe I don't know if they need to interact with another protein. So something that we can do is to put one region that we know that is able, this is the byte that we know that are able to interact with another protein that is a prey. And we join this stuff, we put together this stuff in the construction. So we have protein DB that I don't know nothing, another that is AD that I know something, and I want to know if they are interacting. So that means that if they are able to interact, this bite and prey, they will be together also. And the very nice stuff is that if they are together, if two proteins are together, these two regions are able to detect two promoters that are separated in the DNA. So if they are alone, they join just one promoter. And if the another protein is alone, they are able to join just to another promoter. Promoter. So the problem is that if they are alone, you will not see the expression of the gene that they are turned on, that in this case is a reporter, can be GFP or can be LAC. So you will detect expression of this reporter just when they are together, because they are in the same moment. At the same moment, they are in both um, promoters, and in this moment, they start the transcription of the reporter. And you can see, for example, if you are using GFP, you can see in the microscope that they are interacting. And this interaction is in yeast. You transform both, you put both construction in yeast, and you can see in the plate some yeast that are with GFP, expressing GFP. And after that, you can do that in massive way with all of the proteins that you have in the genome, and you can ask direct interaction with all of them. So, but particularly in GIS2 hybrid, usually we run super big and massive experiment with many transformation pairwise, two proteins interacting per a matrix of different Gs that we can select, and we can check if they are expressing or not a reporter, and we can conclude that we have interaction or not. So at the end, you finish like a matrix where you can say, okay, this protein is interacting with this one. Yes, it's a 1. This protein is interacting with another one. Is no, 0. And you can start to build this big matrix with all of the information about the interaction. Do you have question here? Another kind of experiment that is very interesting for this stuff is the proximity ligation enzyme that is similar to hybrid, but this one is is super important for example, when we can evaluate interaction in just two proteins that we can know or we want to know if they are close working together. And here you can allow a distance between the antibody that you can use in, and if they are together, they can produce fluorometer, and you can detect this interaction also watching fluorescence reactions in microscope. That is very, very cool. For example, one of them is a fish experiment. I don't know that you know about this experiment. Fish. It's very nice to describe interaction in proteins. Do you have questions here? Comments? Okay, but you can see here that we are just trying to look what is the connection between our nodes, you know? This is just watching if we have connection or not connection. So at the end, we can finish with this kind of stuff. When we are checking protein interaction, we can say, for example, okay, this one is binding to this one, and this one is binding to this one, or this one is binding to this. Or for example, this system is completely separate to the rest of the network. So that is very cool because now we have, for example, we can know, okay, this is the prey, and this one is connecting with this one, and they start to work together when they are in the cell, because we can see this evaluation, for example, GIS2 hybrid or another experiment. So at the end, this network comes from this information. After that, we create a matrix, and after that, we create this kind of representation. And here you can see some specific changes in the network. For example, when you are not finding, for example, many connections, you can finish with this kind of nodes that are disconnected. That means that they are not together with another proteins. This is super common, and when you will see some networking publications, you will see many nodes that are alone. That means that they are not, at least they are not connected by the stuff that we are evaluating, you know? For example, here maybe they are connected like a co-expression, but maybe they are not connected by if they are having contact with another protein. So this is a layer of information. We can have another layers of information. Now we will talk about metabolic networks. As we commented before, metabolic networks come from the reaction that are by enzymes, catalyzed by enzymes. and they started with some product, some compounds, and they produce some product. And we have some information here, like for example, allosteric control when they are competing with some compounds, or also we can see what regions is starting after different another region, like they are sharing some metabolites, and another metabolite is happy here, and they cross to another enzyme, and another enzyme producing another metabolite, and this kind of network could be like a cycle, that is completed close, or like a one network that is starting with some products and finishing with another products, like a open network. So what kind of plot we can see, or what kind of graph we can see in these reactions? We will have nodes with that represent substance and hyper-H reactions, and these kind of plots has the entrance of this kind of plots is the substance, and the final part, or the output, is the reaction, final reaction of the nodes. And we can see, for example, some substance like a substances like ATP, ADP, water, and we can see this kind of stuff information in that network. So, for example, you can see some of these examples in these databases, and we will see some of them in our next class. For example, here, this is a metabolic network where you can see, for example, one protein that is starting one reaction, and this reaction is stopping this reaction. And we can see, for example, different representation like a compounds, activation, inhibition, expression, repression, indirect effect. And we can see, for example, phosphorylation, dephosphorylation, and all of them are different representation of H's in this kind of networks of metabolic reactions. This is the main way that we show metabolic reactions, like showing the different steps of the reactions. For example, if we start with glucose, and after that a phosphorylation happen, and maybe this change the structure of the glucose, and after that we can see how they transform in another molecule. This is a classic way to show metabolic pathways. But in terms of metabolic networks, we can show different kind of information. For example, we can show super complex network with all of the steps of all of the metabolites and all of the substrates. But also we can show just reaction, just compounds and enzymes, or even just compounds or just enzymes. All of them are networks. All of them show something interesting, and we can understand many stuff. For example, in genetics, usually we use this kind of networks where we are checking the position of the enzymes in the different part of the reactions. So usually we are trying to represent this kind of network using some codes like a AC number. Do you have idea about AC number?
It's
And
the
where?
enzyme classification.
Exactly. And it's directly to the function, right? It's directly, we can know, for example, if some enzyme is a polymerase or protease or whatever, right? Another part is about that's called substance, and reversibility clearly indicate, because if you remember, all of the reaction can be in reverse. In metabolic pathways, we can see, for example, one reaction that is happening in one direction and also can happen in another direction, and usually we can show that in the pathway. So as I mentioned, there are some pathways that are open and some of them are closed. That depends on what kind of reaction we are looking for, or it depends on what kind of reaction we are showing in the plot. You have a question here? Okay. We can see metabolic pathway as a like a the organization that can happen during the reaction, like something like without real order, or we can put in hierarchical stuff, and that is very clear that some compounds can be happen just after one reaction, and we can show this kind of reaction in hierarchical way. So we were discussing about some characteristics that I showed you in the first part about talking about networks, but we need to talk about informal way the emergent properties of the biological networks. There are many kind of networks. Social network, for example, is a kind of network. Electrical network, for example, in a city is a network also. So many of them have different kind of rules, different kind of properties. But biological network has super specific kind of properties. For example, biological network are scale-free topology. So some of them are super central network with super high connectivity, and some of them are alone, as I show you. Some of them are super separate of the rest of the body of the network, and some of them are super connected. This is a scale-free topology. That is a property of biological networks. Another property is a small world property. That is very cool property. Do you remember about this small world property? Maybe you know that in social media sometimes somebody talk about la teoría del mundo pequeño. You know that? This is I will show you something about that, but this is an experiment, social experiment, that the people start to see. For example, do you think that is the rule said, that the law said, in what small world property network, all of the nodes are connected to the another node in the network with super small steps, surprising small steps. For example, one of the stuff that the people start to see is that, for example, Daniel is 6 or less steps separate of Paul McCartney. Because maybe the father of Daniel has a friend that is living in US, and this guy has a child in the school, and the father of this child is in the Hollywood cinema, whatever, and they have contact and they are friends. So maybe they are close to 4 or 6 steps, and they started to see that at least in this kind of network, many nodes that could be, you can think, okay, we are super separated of another nodes. In this property means that is not true. We are super close. For example, in human, that is true. In social human society, it's true. We are super close between us, and we follow the small world property. And another property of the biological network is a modularity or robustness. That means that they have many connection in one point, or some of them are not super important. That means nothing because we have like a robustness in the network. For example, here just is a sample about Facebook. Do you remember? Maybe you don't remember, but Facebook long time ago, maybe in eight, something like that. They produce this kind of network using the information of Facebook. And in this kind of network, you can see you are here, and maybe you are connected with this group, and also you are super connected with this group. But for example, you can find somebody that you never know that this person is connected to you and is part of your network, but maybe you don't know. Maybe you know to another person, and for example, is part of your connection with somebody super important for you, and also is part of this network that is close to you, but you don't know this person, but is super connected to you because many people is sharing connection. You know, that means that at least in social stuff, this kind of network is happening also. There is at least 6 degrees of separation. And this is the guy that showed that, Stanley Milgram. He's a social guy, he's a social scientist. That was super cool. And we found that this is the same rule that follow the biological networks. So now we can conclude that all of these networks are interconnected, and we can understand phenotypes, and we can understand these emergent properties using all of these networks. They are not disconnected. It's clear, right? So now we will start to understand a little bit more about what happened in maths about the networks. So as we were talking, you can see this is the connection, for example, of one gene that is activating something like a transcription factor that is activating some genes, or a repressor that is decreasing the expression of one gene. And we can describe that with nodes, right? Like a X that is activating Y, or X that is desactivating Y. And this kind of connection can be used to build this kind of matrix. Like, for example, here in this simple network, you can say X is activating Y. So X in interaction with Y, this is a real interaction and positive interaction. And probably X is using a mechanism of autoregulation, like a feedback, for example. And this is Y, that is a feedback alone. So this regulation Y to Y is a negative interaction. And also, Y can interact with C, and they have both directions. Just you can put these directions here. So you can see that we are starting to build this matrix that we can use to build a network. And at the end, we can finish with this kind of stuff. Like all of them are super connected, and many of them are not super connected, and some of them are part of this hair ball that is super typical in biology. And as we were watching, we can put this kind of stuff, like we can put this kind of representation, but also we can put this kind of representation that is a little bit more simple, or even we can use this kind of representation, decreasing the complexity of the model. So what properties we can have in the networks? For example, one of them is the degree of the node. The symbol of this stuff is K. And for example, here talking about this node, A node, we can count how many connections they have. They have 1, 2, 3, 4, and 5. So the connectivity of this gene is 5. This is one of the properties that we are measuring in our networks. Another property is, if we have direction, we can say if something is in or out. In this case, we can see that one of them is out. So we can describe this kind of stuff in the equation and put this kind of distribution of the degrees for each node. So at the end, we can finish with this kind of summary of the information. For example, if I ask, what is the node that has more connection? For example, it's this one. How many of them has this amount of connection? Not many of them, right? They are here, but it's just one. And another one, for example, this one has 5. How many have 5? Maybe 2, maybe 3. So we can start to find these interactions and with this kind of maths associated to the network. And at the end, we can have something like that, right? The fraction of nodes that have some connectivity and how many of these nodes have this connectivity. And this is the log fraction. And you need to know that many of these biological networks follow the power law. So when we are watching characteristics of the biological network, usually they will follow the power law, and we can see something like that. Okay, there are some networks that are random networks. For example, this is not a biological network. You need to see something like that. At least after this class, you need to see something like that, and you need to say, this is not a biological network. Daniel, why is not a biological network?
I'm guessing that because the distribution is uniform.
Okay. And that can happen in for example, interaction protein-protein?
I wouldn't think so. I'm not sure, but I wouldn't think so because the previous examples that you've shown is that not all the nodes are connected.
Exactly. So it's super impossible to see something like that in biological networks. There's this kind of example, for example, electricity connection or internet, all of them has this kind of connection, right? But it's not a real biological network. So hierarchical network can happen, but also is part more than connectivity, like a telephonic connectivity or something like that, but it's not clear in biological network. But usually we have this kind of scale free with this kind of power law, and this is typically one biological network that we can see in different kind of data. So another metrics that we can define here is the clustering coefficient. That means that not just is forming connection between one node, if not maybe many nodes are close together, are close between them. And this is forming a cluster. And this clustering coefficient is very important to describe, for example, sometimes if some of them are super close, maybe they are working in something similar. That is very common in biology. For example, transcription process usually is happening like a cluster, and this cluster is forming something new, and this is the real function together. Do you have question here? Okay, this is clustering coefficient, and we calculate these clustering coefficients when we know how many neighbors has each node, and we summarize all of the neighbor numbers, we can create this coefficient clustering. And it's something like that. E here is connection between the nodes, and K is how many neighbors they are. For example, here, can you tell me how many neighbors has A node?
Does it have to be like direct neighbors or
Yeah,
all of
direct network. Okay, the concept of neighbor is direct connection with nodes.
So 4.
Okay, 4. They have 4. They have 5, right?
Oh yeah, 5.
5 neighborhoods. So neighbors. So that means that we can calculate the clustering coefficient using these numbers. Okay, that is cool for this small network, but maybe if we have an enormous network with all of the genes in one genome, could be super difficult. For the reason we have many programs that we can use to evaluate this kind of stuff. Even we can know what is the clustering coefficient, what is the average of degree in the network. So these properties, usually we use some programs to know this information. Um, Give me. Can you see my screen? Give me 1 minute. Can you hear me? Okay. Can you see my screen? Yes. Perfect. So, okay, we know what is the degree. That is the number of connection, the number of connection that they have. We can know what is the clustering coefficient, calculating how many neighbors they have. This is per number of nodes. And we can see that some of these are loops or feedbacks. And using what is the average of connection between one node in the border of the network and another nodes in another part of the network, we can calculate what is the diameter of that network. And we are looking the shortest path between the two most distant nodes. So that can be 3, 4, 6, 11 nodes. And this distance is the property diameter. So when somebody asks you, what is the diameter of this node, this network, you can calculate what is the short path between the two most distant nodes, and you can quantify how many nodes they are separate this path. This is another emergent property. So now, what is the robust? Why we say that the network are robust? Because many of the nodes, many of them, even more than 90% of the nodes, if we remove these nodes, if we delay the node, we will not see one effect, or we will not see super drastic effect. But there are some nodes that are hubs. The hubs are central nodes with super high centrality. That means that they are connected with many nodes. And calculating the average of connection in the whole network, we can describe what genes are hubs or what nodes are hubs in our network. And that means that these hubs are super vulnerable targets if we remove this element. For example, you know that many genes, when you produce a mutant in experiment like classical genetic experiment, that we delete one gene, usually we cannot see phenotype. And that means maybe it's because this is a no central node, it's not a hop, could be super disconnected gene that is important for something, but maybe not specific for our phenotype or our stuff that we are looking for. And we don't have phenotype. And this is very classic in genetics. But if you look for hops and you remove the hops, it's probably that you will have a super drastic um, changes. The problem is that usually the hops maybe, or in terms of biology, in terms of genetics, can be some essential genes, like some genes that are super important to hold the housekeeping genes. So we need to see when we are designing experiment to evaluate networks, we need to think in all of this stuff. But this is a property, robustness is a property of the networks, and it's about that many of the nodes are not super important in terms of phenotypes. So, and another stuff is that is a particular thinking in biological networks that we have some regions that are able to activate another regions. For example, here I am summarizing here many properties of the biological networks. The centrality, that is identify important nodes in the network, describing hops. The robustness, that is how many how resistant is the network to changes if we remove something. The clustering property, that is how they are in local grouping, the nodes. The path length, that is describing how wide the distance between two nodes. And from this property come from the diameter of the network. Also, we can talk about the small world, that is this stuff that is they are separated with super short path between two nodes, and they are scale free. That means that some of them are dominate, some not are dominate, and some of them are not super connected. And you can see here example of network. For example, random network is not a biological network because we know that some of them are super important, like for example, in the case of genes, and also it's not a regular network, but because they are not all of them connected, and it's something like this one. And another stuff is that we can see these hubs that are connected between them, and they are forming clusters like that. For example, here you can see in this simple network, we can talk about three different clusters. This is one cluster, this is another cluster, and this is another cluster around some hubs that are central in our network. So you have questions here? We will start with the last part that is about gene expression mechanism, regulation, and patterns. Comments? Everything is clear? More or less? Super fast? Slow? Okay, perfect. Okay, we will talk about gene expression. As we mentioned, we will have expression of genes regulated by transcription factors, and this transcription process will produce mRNAs. And these mRNAs, we're able to read these mRNAs, right? We will see sequencing, for example, the amount of reads of the mRNAs, and all of these mRNAs, the amount of mRNAs, and how they are producing proteins are regulated by chromatin epigenetic changes. Do anybody has idea about what mechanism of epigenetic mechanism? What mechanism that you remember? chromatin regulation. Any idea?
DNA methylation.
Exactly, DNA methylation. And what is the phenotype or the chain that you can see? For example, if you have methylations, what is the effect of the methylation?
I don't know if I'm remembering well, but I believe that it makes the DNA more compact.
Exactly. This is about exactly. It's closer because you are having methylation in, probably there are two kind of methylation or two kind of modification in the chromatin, in the histones or in the C base, cytosine. So that main process to regulate expression is changing the chromatin axis by epigenetic changes or signals like methylations, acetylations, you know. Another kind of control is transcription factors. When they are turned on, they promote the transcription using promoters regions, enhancers also, silencers also, that are another elements in the DNA that these kind of proteins are able to read and to start the transcription. But there are another kind of modification like a post-transcriptional modification, like a mRNA splicing, an alternative splicing, capping, polyadenylation, stabilization using regulate which mRNA reaches of the ribosomes. There are some changes also using small RNAs, like a microRNAs, promoting the degradation of the RNA or sRNAs. Translational process using microRNAs also blocking the ribosome, and post-translational with some modification in the protein. So all of these changes are reflecting in our results in the network. What kind of process are happening? Transcription by RNA polymerase 2. We can see elongation, for example, process that sometimes is about the template of the DNA processing to create the mRNA. Co-transcriptional processing, this is kind of process that are happening. An alternative splicing, that is forming protein isoforms. We have introns in the genes, and after that splicing, we lose these introns and we form the mature mRNA. And this is but there are alternative splicing, for example, where we can find different isoforms of the same gene. So that is very important, and is good to have the idea about these kind of elements. So at the end, we can have different networks. We can work with transcription factors that are super important, and we can detect the transcription factor in the network and to say, okay, this node, this element in the network is super important, it's a transcription factor, and we can give a weight because they are regulators. They are master regulators. They are able to modify the interaction. They have autoregulation, and they have combinatorial control. So for this reason, sometimes when we are running analysis, preparing network, we need to detect the transcription factors and to say this node is super important because it's a transcription factor that can change the topology of the network. There are another kind of regulators that are super important in this kind of regulation gene networks. Do you know about that? This is very important for you, and you need to know about this stuff. Ana Karina, I know that you know. Tell me
About what exactly?
about these elements. They are regulated element. Are
I
super
mean,
important for regulation. Sorry?
I don't know a lot about them. I've heard about them, but I know that they just like interfere or silence genes, as Daniel said previously. For example, the microRNA and the small interfering RNA, I've heard that they.
Okay, okay. And there are another kind. Do you know about this stuff?
I know that they have something to do with also regulatory stuff because a friend of mine is actually working with, I think, the long known coding RNA, but I'm really not that familiar with
Yeah,
with
that
what
is
they do.
it's very cool because they are also regulatory elements and they are super important. Now we know they are super important because previously, I don't know that you remember when the people start that project of human genome, they start to see that just 2% of the genome is able to produce protein. And the rest of the genome is no codifying nothing. And the people in the first part start to think, this is a chunk of the DNA. It's a region that are no important because they are not producing protein. But now we know that many of the regulators from the expression or regulator of the expression are codifying in these regions. And long non-coding RNAs are RNAs similar to mRNAs, but they have a lot of stop codons across the body of the messenger. But sometimes, for example, they can have the behavior like a sponge, and they reduce the expression of the mRNAs, or for example, they catch microRNAs and they are like reducing the regulation by microRNAs because the long coding RNAs is like a buffering effect, you know. The real gene that the microRNAs are able to control are expressed, but at the same time, sometimes the long coding are expressed, and they have some regions are similar to the real mRNAs, and they reduce the effect of the microRNAs because they are present, like a competing for the microRNAs. That is very cool regulation, and they are super important. We know now that, for example, during the development, in the embryonic development, the long non-coding RNAs are super important. And they are working, for example, remodeling chromatin, transcriptional regulation, and in splicing. Another kind of RNA that are regulated are the ribosome RNAs and tRNAs. Also are important for regulate. And sometimes in some networks, we need to take account these elements. At least now it's super common to see these kind of elements in the network. You can see genes, but also you can see some long non-codings, RNAs that are detected, and we can see co-expression in the genome. So this kind of the architecture of this network as showing all of the H's that are regulatory interaction, and if they have motif are showing in the network, and also if they have autoregulation, we can show like a loop, and we can show also if the transcription factor is regulated 2 or more genes. That is something that can happen. How we can know the expression of the genes? Because we sequencing, usually we are sequencing now. Previously, we run microarrays. The microarrays was super important for a long time because they are super cool because we have defined what genes we want to evaluate. For example, 2,000 genes. And we developed very nice chips of microRNAs for E. coli, for yeast, for humans, for rats, for mouse. So there are many good stuff here in microRNAs, but now we are using next generation sequencing because it's cheaper, because it's super nice resolution, because we can know long non-coding RNAs, we can see changes in the ribosomes. So that is very informative, cheaper. And here I am showing the NovaSeq 6000 that during your career you will see that because it's super important for everybody now. And also we can have single cell RNA seq that is also with this information we can create gene expression networks. We can have a spatial transcriptomic, and we have a tag-seq that is about what regions are open for transcription to see if we have a chromatin modification and what genes are interacting with a specific region of the genome. So with this information, we try to understand how they are changing together. And this is a joke because the people say that it's like if you saw somebody that is running after they stole a store and behind of them is running another person, by association, you can say all of them are friends, all of them are saying the same and doing the same. And this is the idea behind of gene expression patterns and network in co-expression networks. That is, if one gene is up, for example here, and another gene is up again, and another gene is up in many conditions, and they are follow the same behavior across many conditions. For example, imagine that each position here is a condition. You will have a cluster and you will say, okay, these genes are together, you know, because they are having the same behavior. And you can conclude they are working together. And this one that is super noisy, maybe they are together, but not super connected, that right? And for example, we can have a gene that is alone. For example, imagine this one, is this one that is here. Maybe this one is not connected. And we can describe this disconnection as a node without connection with another one. So here we need to calculate many stuff, right? We need to calculate a correlation using Pearson or another kind of correlation models. And this model can say, okay, it's a negative correlation or it's a positive correlation. So that is very important for this kind of networks. And at the end, we can build, using the pattern of expression, we can build our networks with many connections and to show that. So in this kind of networks, there are some stuff that are super important, like if some of the genes are transcription factors, if some of the genes are come from some different organelles. For example, some of them are expressed in nucleus or cytoplasm. You can put this information also in these kind of networks. And that this is tissue specific is super important because if you have a liver, brain, muscle, they have different patterns, right? And you need to describe these patterns and create this network for a specific tissue. And also now, single cell, this is a new technology, is super important because we can see expression for each cell. And at the end, we can have a very nice networks interacting between them and to see new properties. Do you have question here? Everything is clear? Okay, so the next class, we will start to work with co-expression analysis. We will have a table, a matrix of expression that come from RNA-seq, and we will try to see and we will try to find these patterns and to create our network and evaluate properties. It's okay? Okay, if you don't have another questions, I think that we can stop here our class, and the next class we can start with R. I need that you try to install R, but if somebody has problems, we can do that in the class. We have enough time to start installation of the R, and after that to look for the package. Also, if you can try to install Cytoscape, but if not, maybe we can run also here in the class. Any question? Okay, everything good? Okay, I will finish the recording process. Let me stop sharing.